Hello, everyone. This is our last set of notes for this unit. So congratulations, we've made it through an entire unit. I'm really proud of you guys. Hang in there. And this is it. This is the last one. So here we go. Today we're going to talk about sex-linked traits, polygenic traits, and we're also going to talk about multiple alleles, which falls in here, although I didn't list it, did I? But we are going to talk about multiple alleles as well. All right, so we're going to start with polygenic traits. And this is when there are two or more genes that influence what a trait looks like. When we talked about earlobes, there's just one gene that indicates whether you're going to have attached or unattached earlobes. However, for all of the coloring involved with people, our eye color, our hair color, our skin color, all of those things are influenced by more than one gene. And in fact, our eye coloring is controlled by about 15 genes that we're aware of, and they're still investigating that there might be more. So those are called polygenic traits. Many of our traits do just have one gene, like our earlobes, and whether or not you can roll your tongue. Others have multiple genes that influence what the trait actually looks like in person, so that phenotype. So the examples are, again, the coloring, hair, skin, eyes, all of those things. Our height and weight are also influenced by many genes, not just one. These can also be influenced by our environment. So for height, the environmental influence can be proper nutrition at a young age. There are plenty of records that indicate as a species, human beings are taller today than they were 100 years ago. And most scientists believe that that's due to proper nutrition or more proper nutrition at younger ages. However, we also know that even with proper nutrition, some ethnic groups and people from different regions tend to be taller or shorter. So there is a genetic component to height, but there's also an environmental influence as well. The same thing is true for weight. If you have ever noticed that different body types in terms of weight tend to run in families, that's because genetics has a lot to do with it. It's not just your environment. It's not just about exercise and eating and calories and all of those things. There is a genetic component. However, those are influenced by your environment, the things that you do. But it's not entirely one or the other. My example for this, typically if we were together, we would have done a lab. I tried to think of a way to do it at home, and there just really wasn't. There's no way for you to have the materials that you would need. So let's take a look at this Punnett square. You guys have done dihybrid crosses where we look at two traits, right? Well, this is a trihybrid cross where we're looking at three traits. So if you think of each pair of dots here as being one gene, okay? So there's three genes. If we look at all the possible combinations for the egg cells for mom, there are three genes. So there's the combinations that are possible. And for dad, here are the combinations as well. When we talk about polygenic traits, we have to think about it this way. There are three genes, which means for each one of those genes, there are different alleles. This one is simplified by saying that there is simply a light gene and a dark gene. No, I said that wrong. That there's simply a light allele and a dark allele. So there's only two possibilities. So for all three of the genes, the allele can either be light or dark. And your skin color is influenced. So this is a incomplete dominance. They're both dominant. They both show. So the greater the number of dark alleles that you inherit from your parents, the darker your skin. The more light alleles that you inherit from your parents, the lighter your skin. So if we look at this, if we take one parent and we do kind of the FOIL method, like we did for the dihybrid cross, and we figure out all the possible combinations of these dark and light alleles that they could get in the egg cells, it looks like this. Since they're both heterozygous for all three genes, it would be the same for the sperm cells for dad. So if we look at this, there's a 1 in 8 chance that the sperm cell will look like this, which is all three light alleles. There's a 1 in 8 chance that the egg cell will also have all three light alleles. Now, the chances of those two getting together are 1 in 64. The same is true for the opposite end with the three dark alleles and the three dark alleles here. There's a 1 in 8 chance that the egg cell will have three dark alleles and a 1 in 8 chance that it will have three dark alleles here as well. So the chances of those getting together are also 1 in 64. Now, you notice that here it's one dark allele and two light alleles, one dark allele and two light alleles, one dark and two light. So if you think about this phenotype, right, 2 light and 1 dark, it's actually 3 out of 8. So you can see that these three are colored a little differently. So it gets super complex. You're not going to have to fill out anything like this. I'm just showing you that polygenic traits are very hard to tease out in terms of Punnett squares. It's not so simple. You could have two parents that are heterozygous for both traits that look like this, that end up with a very light-skinned child and a very dark-skinned child. What are the chances of that? Again, 1 out of 64, but it can still happen. So the more dark alleles you inherit, the darker skin color. So there's lots of variation here. And this is just assuming that skin color is controlled by 3 genes. And it would be interesting, you could look it up if you want. I don't know how many genes there are that actually influence skin color, but this is what it would look like if there were 3 genes that influence your skin color. Imagine your eye color. That we know there are at least 15 genes that influence your eye color, and we suspect that there might be more. Can you imagine putting a Punnett square together with 15 different genes? This is just 3 genes. A 15 gene Punnett square would be enormous, right? So predicting the eye color of offspring is a tricky thing. And it doesn't always come out the way you think it will. I'm sure you know examples of families where both the parents have very dark brown eyes and they have a child with, beautiful blue eyes. I mean, it happens. And we're also unsure why eye color can change over time. When I was younger, my eye color was actually much darker. As I've gotten older, there is more green and they are lighter. And when I was little, on all of the health forms, dark brown eyes. Now I put that I have hazel eyes. There's some green and brown in there. So mine have gotten lighter over the years. And scientists are still kind of looking into all of the ins and outs of our coloring, which genetically can change over time. It's interesting, very interesting. So genetics is way more complex than you've been led to believe. That's the bottom line. But polygenic traits mean that there's more than 2 genes that more than 1 gene. There's 2 or more genes that influence the phenotype. So you have to think about all 15 of those genes when you talk about the phenotype of eye color, and it gets really complicated. Don't worry, you won't see anything like this trihybrid cross. So multiple alleles. What if it's just a single gene, right? But what if there's more than 2 options for a trait? Let's look at the fur color of rabbits, which is kind of cool. The brown color of rabbits, and if you see wild rabbits out in your yard, I live here in Bexley and we have rabbits all the time and they're always brown. So we call that the wild type because that brown fur coloring is the dominant allele. And it's dominant over all the other possible alleles. Now, if we were to catch some of those rabbits and we were to selectively breed them, we find out that there are other color combinations that show up, but only if the brown color isn't present. So there's the gray coloring, which we call chinchilla. So they did a lowercase c because it is recessive to the brown coloring. And then they put a little ch to indicate chinchilla. And through testing and breeding, we found out that the chinchilla coloring is dominant over the Himalayan coloring and the pure white albino coloring. So if you were doing a Punnett square with an albino rabbit and a Himalayan rabbit, you would use these two and you would follow the order of dominance here. If there's a Punnett square like this, you would be given all of this information. You wouldn't have to know what's dominant over whatever trait. You would be given all the information necessary to fill in the Punnett square and make predictions about the offspring. So that's multiple alleles in animals. Oftentimes fur coloring and fur patterns, like spots on a dog or like the calico nature of a cat, those are often multiple alleles. And they can show complete dominance like the brown fur, or they can show incomplete dominance where, maybe the brown and the white make a pink rabbit. I don't know. So there's lots of different relationships between those alleles, but there's multiple alleles. The other cool one is human blood types. So let's take a look at that. So first we have to go all the way back to last semester, talking about the cell and the cell membrane. Do you remember the tags on the cell membranes? Well, blood type is an example of those tags. So if you are type A, that means that all of your red blood cells have a little tag that says type A. Not literally, but that's what. So it has the type A tag. And your immune system sees that tag and it's like, hey, all is good. I know this tag. This is me. We're in good shape. If you were to give a person who has type A blood, type B blood, the type B blood means that it has a little tag on it that's type B. If you were to give the type A person the type B blood, this person's blood looks at that tag, this person's immune system looks at that B tag and says, whoa, I don't know what this is. It's an invader. It's dangerous. And a huge immune response is launched. And the immune system can be so aggressive that it's killing blood cells. Now, if the person got a transfusion, then they needed blood for some reason to begin with. They're already sick, or they have been in a car accident, or something is going wrong that they needed the transfusion in the first place. If the body launches a huge immune response on top of that, the result is often death. So it is very important to pay attention to blood type in those situations. If you are type AB, then you have the tags for A and B. If you are type O, this is the recessive trait. So A and B alleles are codominant, but they are both dominant over type O, which is recessive. And essentially that means you don't have any tags at all. This makes type O the universal donor. So if a type A person needs a blood transfusion, they can only get blood from another type A person or a type O person, because this person doesn't have any tags. So when their blood goes into this person, their immune system sees the red blood cell, but it doesn't have any foreign tags on it. So it leaves it alone and everything's fine. That is why people who are type O are called the universal donors. They can give their blood to anyone. Type AB blood, they are considered the universal acceptors. You can put any kind of blood in them and it's fine because their immune system recognizes all of the available tags. So it doesn't matter if the blood type being donated is type A. Hey, that's all good. We recognize type A. If type B blood is donated, hey, it doesn't matter. It's all good. We recognize type B. And if type O is donated, that doesn't matter either because it doesn't have any tags at all to alert the immune system. So here's kind of a visual, like what it would look like if we stylized it. And then the phenotype, we would call it A, B, or AB. The genotype, we do a capital letter I just for the blood. There's the recessive, right, for the O. And then we put a little A and B for codominance. So a capital B and a capital A because they are both dominant. So if someone is AB, they have this blood type. If they are type A, they either have homozygous dominant type A or they are heterozygous with the recessive allele. The same thing is true for type B. All right, if you have questions, let me know. All right, so the last thing on our list here are sex-linked traits. So these are traits that are linked to the sex chromosomes. So here we're talking about chromosomes. So what happens if a trait is on the Y chromosome or the X chromosome? All of the examples that I'm going to give are on the X chromosome. Because if you notice, the females do not have a Y chromosome. So there are some Y-linked traits, but we are not going to talk about those here because it's a whole different sort of Punnett square. It's a whole different looking situation. So all of the examples that I list are X-linked traits, meaning they occur on the X chromosome. Now, some examples that we'll use are hemophilia. This is a blood disorder where your blood cannot clot. Red-green color blindness. So if you're red-green colorblind, then the color red and green look sort of a grayish color and you cannot distinguish between them. This is the reason why red lights are always red, yellow, green. Okay, they're always red on the top. You never mix those up because people who are red-green colorblind go with the order of the colors instead of the color of the colors. So if the light on the top is on, it's a bright gray instead of a plain gray, then they know it's a stop sign. If the bottom one is lit up and it's bright gray because it's lit up, they know that that means go. So you'll always see them in that top-down configuration, or it's left to right. So with the red light on the left and the green light on the right. It's never the other way around to help red-green colorblind people. Another example of a sex-linked trait is muscular dystrophy. This is a disorder having to do with the structure and the strength of the muscles and the formation of your muscles. So I like this little diagram because if you look at the possible combinations of chromosomes, X chromosomes and Y chromosomes, between females and males, you'll see that the sperm cells, so males have an X and a Y chromosome, females have two X chromosomes. You'll notice that the male can produce two different types of sperm cells, those with the Y chromosome and those, with the X chromosomes. The mother can only produce X chromosomes. So she has two X's, so some of her eggs will have this X and some of her eggs will have this one. This explains why we have a 50% chance of having boys and a 50% chance of having girls. So if you pause the video and take a look at this Punnett square, it shows that you end up with 50% boys and 50% girls. So what do we see with sex-linked traits? One of the things that we see is that men are more likely to express recessive disorders that are sex-linked to the X chromosome. That's because the males cannot be carriers. And a carrier is someone who is heterozygous for a recessive disorder. So we're going to use H's. We'll talk about hemophilia, the blood clotting disorder. If there's a female carrier, that means that she has the dominant trait, right? The capital H, but she also has the lowercase h. A female who is a carrier for hemophilia will not actually show the trait of having hemophilia. She may not ever know, especially if there's not a family history, like if this is a new mutation that occurred that resulted in hemophilia. But she may not know that she's even a carrier because She has one dominant trait, which means that her blood can clot, and all is well. We have a male here with one X and one Y chromosome, and he has the dominant trait of not having hemophilia. So these two get married, and we can take a look at the so we're talking about hemophilia. We can take a look at the chances of their offspring having hemophilia, even though neither parent has it. If mom is a carrier, then it is possible for some of their offspring to have hemophilia. Let's take a look at that. So if we fill this in, we put these two H's, capital H's and their X's in the same box. We put this X with its lowercase h along with this X with its capital H. We've got our girls over here, and then our boys over here with an X from mom and a Y from dad, X from mom and Y from dad. We end up with one homozygous dominant daughter, one daughter who is a carrier. We end up with a son who does not have hemophilia and a son that does have hemophilia. So in this scenario, if we have a mom who's a carrier and a dad who does not have the disorder, none of their daughters will show the disorder. They may be carriers like their mother, but they will not have the disorder. On the other hand, the males, if mom is a carrier, half of their boys, there's a 50% chance that any son they have could have the disorder. As a result, in a population, you are more likely to see these disorders, hemophilia, red-green colorblind, and muscular dystrophy in men than you are in women. The only way for women to show the trait is that one of the parents is already affected. So if mom can only donate the recessive trait, then both of their sons will have the disorder. But dad has to have the disorder. So if dad is red-green colorblind in this case, the only way for the daughter to get the red-green colorblind trait is for mom to be a carrier, or for mom to also be red-green colorblind. So if you work a couple of these out in examples in your book, and on the worksheet that was assigned last time, you'll see that it's way more often for boys to show the recessive trait. And that's because they've only got one shot. They don't have the option of being heterozygous. They either have it or they don't. This daughter has the option of being heterozygous because she got one from her dad and her mom. This boy does not. He only got the option of inheriting the allele from his mother. So boys either have it or they don't. They cannot be carriers. And let's see. I think that's all, but I'm not sure why it's not clicking. Let's see. Yep, that's the last one, and I'm hidden. I will see you guys next time. Come to a Zoom meeting and have a great day.